A very good morning to all of you. So today's our topic of discussion is what is the difference between colorimeter and spectrophotometer. This difference is can be asked in a viva as well as for the theory. So you must know the difference between spectrophotometer and colorimeter. Okay. So let's start the topic. First is colorimeter, spectrophotometer. So what first difference is that in colorimeter we detect the solute in colors if detect the solute in colorful solution. Colorimeter, why its name is colori? Because we in colorimeter we can detect the concentration of any solute only in colorful solution. Okay, so for colorimeter detection the solution should be colorful. It cannot be detected in the colorless solution. While in spectrophotometer we can detect the solute in both. That is your colorful as well as colorless. That is your colorful and your colorless solution both. Colorless solution both. Okay. So this is the first difference, that is in colorimeter we will detect the solute in colorful solution only. Okay. While in spectrophotometer we can detect the solute in both colorful as well as in colorless solution. The second difference is that it uses visible range of wavelength only. Okay, it uses visible range. What is visible range? It is around 400 to 700 nanometer. As you know, the electromagnetic radiation. So there is what X-ray, UV ray, visible, infrared, all there. So this colorimeter, it use only visible range of wavelength. Okay. The wavelength which we use in colorimeter is cover only your visible range. That is your 400 to 700 nanometer. While what happened in spectrophotometer, as you know it before, while we have studied the spectrophotometer, we have just divided the spectrophotometer into three types: UV spectrophotometer, visible and infrared, on the basis of your wavelength range. So it used for visible, ultraviolet and infrared wavelength range. Got it? Wavelength range. So what is second difference? The wavelength range in colorimeter is of visible range. That is 400 to 700 nanometer only. While in spectrophotometer you will use visible range, that is 400 to 700 nanometer. You will use UV range wavelength. What is UV range? 180 to 400 nanometer. And infrared. What is infrared range? That is of 700 to 1500 nanometer. Already we have studied while just cover the spectrophotometer. Alright. Now, what is the third difference? The third difference is that it is less expensive. Okay, if we will see the cost wise, then it is less expensive. While this spectrophotometer it is more expensive. Okay, so cost wise, if you have a low budget and if you want to measure the concentration of unknown, then you will prefer colorimeter, because expensive, it is less expensive while it is more expensive as compared to your colorimeter. Okay, next difference is that accuracy. It is less accurate. Colorimeter is less accurate while your spectrophotometer is your more accurate. Now understand why it is less accurate. Because in colorimeter we will detect this solute concentration in colorful solution only. Okay, so for colorimeter detection our solution should be colorful. And for colorful, maybe there is any error while you are performing your experiment and you are not doing any heating process properly, then your color will not come properly. And that color you want to for detection of your OD, and that color didn't generate while you are performing your experiment and while you are heating. So there may be chances of your error. That's why because of using of colorful solution, the accuracy of this colorimeter is less, while a spectrophotometer it is more accurate. Got it? Next is that here filter is used. In colorimeter you will use filter for wavelength. You will use filter for wavelength. Okay. While in spectrophotometer, what you will do? You will use prism. Okay. We will use prism on monochromator. Or monochromator. While we have studied yesterday the instrumentation, we have just drawn the prism. That prism split the light. Okay. So one difference is that in colorimeter you will use filter. And kind of filter you will use for separation of wavelength, while in spectrophotometer you will use prism or monochromator. Okay, so these are the 5 differences between colorimeter and spectrometer. Let's understand once again. Colorimeter detect the solute in colorful solution only. Spectrophotometer use both colorful as well as colorless solution both. In spectrophotometer, we use only visible range of wavelength, that is 400 to 700 nanometer. While in spectrophotometer, we can use visible range, UV range as well as infrared range of wavelength. It is less expensive, while spectrophotometer is what? It is more expensive. Okay. And accuracy, regarding accuracy, colorimeter is less accurate, while your spectrophotometer is more accurate. And in colorimeter, we use filter for wavelength, while in spectrophotometer, we will use the prism or also known as monochromator for splitting the light. So this is the difference. Thank you. And you must read this for the bye.